Hello, everyone, and welcome to today's webinar, Increased Protein Coverage for High-Quality Peptides with a Streamlined, Scalable Sample Prep Workflow. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Covaris. To learn more, please visit our sponsor's website at covaris.com. Now, we encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Additionally, if you experience any technical questions, you can submit them in that Q&A box and we will answer you directly. I would now like to welcome our speakers, Mo Haynes, Senior Research Associate, Broad Proteomics, Broad Institute of MIT and Harvard, and Deb Bhattacharya, VP of Business Development, Emerging Markets at Covaris. For complete biographies on our speakers, visit the Biography tab at the top of your screen. Mo, Deb, welcome. So happy to have you. You may now begin your presentations.
Well, thanks for the nice introduction. Appreciate it. And also thanks to everyone who's attending and who's dialing in. Feel free to drop your questions in. We always love to get questions. So the first part of today's talk, I'm going to talk about streamlined, scalable sample preparation workflow, especially focused on proteins, which is something that is gaining a lot of traction in the market. So without further ado, I'm just going to move on straight away into why we are focusing in the world of protein analysis. What are we doing? How does our technology actually is enabling better sample preparation? So it's nothing new to the world of of proteomics or protein analysis, folks, that regardless of whether you are targeted or untargeted, whether you are looking at global proteins or whether you are looking at a specific protein therapeutic and monitoring its pharmacokinetic, pharmacodynamic behavior, there are a few things that are common that everybody always want to achieve. So best data quality, ensuring that you get highest sensitivity, highest resolution, and good information. Ability to scale, not just in terms of volume of your samples, but also number of samples without spending a huge amount of time. And also understanding that what can be a niche workflow today can be implemented in a regular lab, in a routine lab tomorrow. So no matter where you are, all of these things are something that you are trying to achieve, whether it's reduction of cost per sample, whether it's transitioning from your research to a routine laboratory-based test, or you are developing a particular therapeutic, or you are identifying a new biomarker. So how we are making this world look slightly better, complications, complexities, and challenges addressed with a little more ease comes in the next few slides. So first and foremost, let me introduce our technology. So it's an ultrasonication technology that uses acoustic energy at a specific sub-base frequency, hence very short wavelength, that goes and hits the samples and creates a lot of cavitation bubbles, which in due course of time coalesces, releases an energy that's used for disruption of matrices, homogenization of tissues, and so on and so forth. So for a long time, this technology has been the go-to technology for shearing DNA, and it still is as of today. The main benefits are a couple of things: that the pressure that comes on the sample is very equally distributed, so that you are not necessarily jeopardizing the integrity or the structure of your sensitive, vulnerable molecules, whether it's like RNA or proteins. And the other thing is that you can actually maintain isothermal conditions with this. So there's no heat that's imparted to the sample, despite the fact that you are hitting the sample with so much of energy. So if I look into this, that there's been a long history behind us where it's been the way to cut DNA for DNA sequencing or shear DNA for DNA sequencing. It's been also used in transcriptomics for chromatin shearing, also in the world of RNA quite a lot. But now we are actually focusing on the other side of the world as well, which includes, proteomics. For the given breadth of time in this particular webinar, we are not going to talk about metabolomics or lipidomics, how we are also enabling those areas. But in proteomics, I'm going to highlight some very nice published examples that would actually be very beneficial, perhaps, hopefully, for you to understand how our workflows, our technology can enable every single laboratory working with proteins. And after me, I have got Mo, who's also going to talk and share some of his own data, which I think would be even more impactful. So right off the bat, there are 3 stages in which we have divided our workflow, especially when it comes to proteins. So if there is one thing that I would love for each of you to remember today, is that this technology helps you address complex matrices and get high quality analytes out of it. So the first stage is where you start with, whether it's tissues or FFPE samples, cells, mammalian cells, organoids. You can take any one of those matrices and you can drop them in our plates or in our tubes, and with the energy, we can figure out how to actually homogenize that matrix, disrupt that matrix to maximize the total yield of proteins that you're going to get or get out of it in a very reliable, reproducible manner. From that point of time, our technology also helps with protein aggregation capture, as an example, in purifying, whether you are doing the pull down, and last but not the least, we have options where we can also expedite the whole digestion process by with our energy, especially if it's on beat digestion, we can accelerate the whole process. And I will explain this a little later, that how we have taken the overnight digestion and brought it down to something like 90 minutes or 2 hours, although you can also still continue to do the overnight digestion. So the next part that I would love to highlight is focusing on different matrices. So you can see cells, fresh frozen tissues, FFPE, which are the most obvious complex matrices that everybody talks about, everybody wants to address. And we have in here different in the boxes right below. You can see the number of cells that we can address. And then if you have very high concentration of cells, 1 million and above, it moves to a different consumable, a different workflow. But we do have workflows for different number of cells, different amounts of fresh frozen tissues, and also FFPE scrolls. So at the end of the day, what I would love to highlight is that we have set workflows, and towards the later part of the presentation, I will also talk about some of the kits that we have launched, that we are in the process of launching. What we are trying to do over here also is making things in a very customizable, because no 2 proteins are same. Every single laboratory, wherever you are, your proteomic assays are focused on different groups of proteins, different technologies, different focus points. So what we are trying to do is to give you a very high starting ground, and then from there on, we can actually work with you to make it very customizable, optimal for your specific experiments. So as an example, here is published some of the publications that I'm going to go over. So this came out of Cedars-Sinai from Jenny Van Eyck's group, where they looked into doing the proteomic analysis for proteins and peptide IDs across thousands of samples using different tissue types. And they have actually created a total curve of the total amount of proteins that they generated, and also between, segregated between, low abundance, medium abundance, high abundance proteins. And they are also using over here a dual trap single column configuration, which along with the AFA-based workflow that we have enabled, along and the mass spec assays, makes it very, very fast for them to analyze their protein samples. So the next part is something that is very close to my heart, which is like FFPE. And I'll again get back to it a little later in the presentation, that this actually was a collaboration work that was with Matthias Mann's group, and they focused on extracting samples or extracting proteins from FFPE samples, and then automating the whole workflow where they extracted with our technology proteins across different types of matrices, which looked into pig liver, heart, muscle, and also compared with HeLa cells, and also showed remarkable high reproducibility when it comes to its peptide depth as well. So moving on, some of the things that I have already mentioned is that we have certain process of using this particular energy in expediting the whole workflow. And this is a classic example where we have a workflow that we have developed and we've published as well, where we took the whole TMT workflow and brought it down to something around 6 hours, but enabled by the AFA technology. So essentially, most of your reagents and workflows continue to be what it is today, and enabled by the whole AFA-based workflow, we expedite the whole process, which gives you that the luxury of completing your whole workflow within 6 hours. So when it comes to FFPE, my favorite topic, and I just alluded that a couple of slides back, what I would love to highlight over here is the critical challenge for those of you who are working with FFPE samples or are considering to work with FFPE samples. So the first big obstacle is actually deparaffinization. And most often, the workflows, the options that exist out there include actually using harsh chemicals for deparaffinization, which not only is kind of dangerous to work with, it also reduces your scalability capabilities. So it takes time. You have to get to the fume hood. And not only that, it often degrades the surface of the tissues, at least, that's embedded in it, which means it's also compromising the integrity of the proteins that you're trying to extract. Hence, for trace abundant proteins, you are unlikely to get enough of it out so that your mass spec sensitivity is good enough to catch it. So what we are doing over here, there are a couple of ways, but this is the only technology, or AFA is the only technology, along with our workflow, that can actually deparaffinize an FFPE sample or a scroll without use of any harsh chemicals. So much so that it can be automated completely. And as such, on the genomic side, there are quite a few of our existing users who are using our workflow for automatically deparaffinizing and extraction of total nucleic acid, including DNA and RNA at the same time. So we are taking the same first approach over here in deparaffinization with our deparaffinization buffer, that we can start with one scroll of 10 micron thickness that can give you enough proteins and hence enough peptides for your eventually bottom-up analysis. So you can start with the deparaffinization, and in the same plate or in the same tube, you can continue to have your next steps, which is extraction of proteins, purification of proteins with beads, such as the protein aggregation capture or PAC protocol, followed by digestion. So that's using a full scroll, but what if you have even smaller samples, if you are heading towards spatial, if you have laser capture microdissection? So this publication came out as part of a collaborative venture with Mayo Clinic last year. So what they are doing right over there is what they did was start with an FFPE block, but they used LCM, which dropped the samples straight away into our 96 well plates. And from there on, we homogenize the tissues, resulting in extraction of proteins, followed by purification or pull down, and on beat digestion. So as you can see towards the lower right of the screen, the yellow bars where is what the standard workflows, as long as it took. Like it was more than 20 hours was how long it was taking them. And it came down to somewhere around 3 hours for the whole workflow. So it's not just about getting quality samples, quality analyzed, but it's also about the ability to scaling it up, to increasing the throughput. So, it's actually quite nice that today, within a very short period of time that we have been focused in the world of protein analysis, we actually have a lot of robust workflows that are not only tested internally, there are quite a few users around the world who are regularly using these workflows, whether it's cells of different number of cells, fresh frozen tissues, or FFPE samples. So all of these together, along with our instruments, we have been supporting different kind of protein laboratories, whether it's academic research, whether it's biopharma, or whether it's a contract research organization, whether it's a clinical research environment. And we also have very recently launched our first kit in the world of proteins, which is starting with cell lysis kit. So the TruePrep protein kit works with mammalian cells, and it is a comprehensive kit that enables lysis, extraction, and digestion. The digestion part can be done with your choice of enzymes, not necessarily that we would provide you with an enzyme. So you can continue to use your enzymes, but use our workflow with our instrument and our kit to expedite, make your process a lot more efficient than what it is. Some of the results early stage that I would love to highlight is that you can see over here the reproducibility, which remains very consistent both for total proteins as well as for peptide yield using this particular kit. So there's very minimal sample handling. You do not necessarily have to consider any transfer steps. Everything's happening in the same strip or in a 9 to 6 well plate format. And the reproducibility has always been less than 15% for proteins and peptide yields. So, which is extremely beneficial whether you are trying to standardize a workflow or optimize the condition. Next up, I would love to highlight, the flexibility or the scalability. So as I mentioned in the Mayo Clinic publication, it's similarly with the cells. We also have that the standard workflow took as long as 18 hours, whereas with our AFA-enabled workflow with the kit, we are down to 4.5 hours. So you can start with as low as 1,000 cells or go up as much as 500,000 cells, and you can still make it scalable because your whole workflow is actually incredibly faster compared to what you are normally used to. So somewhere to the tune of 4 times faster. So whether you consider it 4 times faster or 4 times increased throughput, that's completely up to you, but we can surely enable that. Thereafter, one other thing that I would love to highlight is that typically we do not recommend and do not use a lot of detergent because your LCMS instruments are not necessarily very kind, or detergents are not very kind to the LCs. So this is another reason why we have a very robust cleanup process for the downstream analysis that also takes care of any even trace amount of detergent. And so there is consistently low levels of residual SDS carryover so that you do not necessarily have to worry about your LC performance even before your sample gets to the mass spec. And thereafter, there are cost per sample. So we, not only in terms of time savings, there's also financial savings because there are a lot of ways you can actually increase your scalability, your throughput, your total capacity of working with different kinds of mammalian cells, different numbers of mammalian cells, and eventually resulting in lower cost per sample. So in a nutshell, this is how the TruePrep protein kit workflow is for the cells, where you start with, you harvest the cells, you start with an AFA in lysing the cells, and then you follow it up with cell lysis and DNA shearing, followed by reduction and alkylation steps, which you do not need to use any AFA for that, but it can happen in the same strip format that we are providing, followed by purification or your pull down, and then accelerated digestion. We also have a workflow in the process of being converted to a kit, but we have this workflow that we have been supporting that I already mentioned to you when it comes to FFPE samples. The similar approach, but it has a couple of more steps additional because you have to deparaffinize, followed by tissue homogenization. Then you have to do the whole reduction alkylation, followed by purification with the PAC protocol, and then the digestion. So all of these things are in the bag. We have, as I mentioned, there are 3 distinct stages that we have focused on in a protein sample prep capability. So from extraction to purification to digestion, our AFA class-leading technology, along with our workflows for proteins, and in certain cases supported by our kits, can enable not only maximizing the total protein yield and peptide conversion or peptide depth from your perspective, but also doing so in a very reliable, reproducible format so that you can be rest assured that you didn't do anything wrong in your sample preparation. And you can focus on where you need to focus on, which is analyzing the LCMS data. And that too, you can do the whole sample preparation faster than ever before. What you've been doing until now, overnight digestion can be avoided if you would love to do so. So to summarize everything before I pass on the baton to Mo, is that if there's one thing, once again, I would love for each of you to remember from my talk today, is that complex samples to high quality analytes. This is where our technology becomes extremely beneficial, extremely helpful, a big enabler to every single laboratory. Thereafter, the protein analysis workflows, whether you're talking about extraction of proteins from complex matrices, to purification of proteins, to expedited digestion should you need that, all of those steps can be easily enabled in a customizable workflow format, depending on what proteins you are going after, what is your sample type that you're starting with, your nature of your matrix, whether it's fresh frozen tissue or FFPE or cells or organoids, and what type of proteins that you are primarily focusing on. The TruePrep protein kit, I talked about the cells. There would be something on the FFPE side also soon that you would be hearing about. All of these things are enabled or are designed to create a streamlined process in your laboratory, which you can further customize to cater to your specific needs. So once again, complex samples to high quality analytes, and that's where our AFA-based workflow for proteins and in general protein analysis becomes extremely, extremely useful. So I don't want to continue any further because there's someone whose talk I'm actually eagerly looking forward to listen to. So without further ado, next up is Mo.
Thanks, Deb, for the introduction. I'm very excited today to show our new FFPE workflow that we've incorporated Covaris into this workflow to process FFPE tissue and to go over all the details in a systematic way. Just a little bit about the proteomics group at the Broad Institute before I get started. We do a lot of protein interactions evaluations. We're interested in what proteins are interacting together, what signaling pathways are being activated. And we're also looking into the PTM landscape. We're mostly interested in ubiquitination. We're interested in phosphorylation, acetylation, and even glycosylation. Another thing that we're also interested in at the Broad Institute is the multiomic and proteogenomic analyses of different disease states. And we do HTX mass spectrometry, cross-linking mass spectrometry to understand more about protein dynamics and drug interactions, And we also do a lot of chemical proteomics. And this gets me to my next point, which is FFPE samples. As we know, FFPE samples has long been the standard for tissue embedding, and it presents an invaluable resource for target of biomarker discovery. Right after surgical excision or any biopsy, tissue gets excised, and then it gets placed in formalin, which would harden the tissue. And after that, it gets embedded in a paraffin wax, giving us a block that looks something similar to the picture on the left side. And these blocks are very stable at room temperature, and that allows any analyst to be able to analyze the same tissue multiple times by scrolling a slice out of it, and then they would carry on with whatever assay they have to run, such as histostaining or any kind of proteomic analysis. And the issue with FFPE samples is that they can be stored for many years. And one question remains is how stable are they? But also for a proteomic application, there might be some hurdles, and that's because we have to deal with wax material and we need efficient decross-linking, as we know that formalin can diffuse through the tissue and can form a methylol intermediate that is not stable. And from that intermediate would end up with a shift base that's able to cross-link between two nucleophilic groups, whether it's DNA or protein. So efficient decross-linking of our tissue is really critical to get good depth of FFPE material. And in a typical proteomics application, we start with a sample, regardless of where it's coming from, whether it's tissue, whether it's any fluid. The idea is to lyse this material, and then we'd extract the proteins after they've been denatured. And then these proteins get digested into smaller peptides. The peptides gets cleaned up, and then they get injected on an LC-MS setup that allows us to sequence these peptides and trace them back to the proteins that they've originated from. And mass spectrometry is known to be very sensitive to many different samples. However, for FFPE samples, that will require some additional processing steps, as we know that a lot of upstream contaminants need to be eliminated, such as wax. Otherwise, we're going to end up with a lot of issues on the mass spec side, which can affect ID, sensitivity, and even peptide recovery. Prior FFPE workflows rely on xylene, out of which, starting from an FFPE block, a scroll is taken out, and then it gets de-waxed using xylene, which is a manual step. And after that, it gets rehydrated with an ethanol water wash multiple times. And after that, the tissue is kept to be dried out, and then it enters into a lysis buffer, and that's followed by decross-linking, and then manual homogenization with a probe sonicator. And then we carry on our typical LC-MS proteomic application of reduction, alkylation, and digestion, cleanup, and then mass spectrometry. And as we see here, this process is really lengthy. And if you want to apply this to a clinical setting, you'll have to process each sample individually. And a higher throughput process is needed to allow for biomarker discovery in a large cohort experiment. So that's where the Covaris comes in to help us with simplifying the FFPE workflow. What we've done first is we've worked with colorectal and breast cancer blocks, and we've done the microtome slicing, out of which we've started at 10 micron scrolls. And these scrolls are transferred immediately into a Covaris plate containing the tissue lysis buffer. And then the Covaris plate is immediately transferred into the ultrasonicator for deparaffinization. This process takes about an hour, followed by decross-linking for 90 minutes at 90 C in a thermal cycler, and then with another round of ultrasonication to homogenize all tissue that's been decross-linked. And then we follow this up by two main proteomic applications to clean up the sample. First, we reduce and alkylate, and then we tested either the sp3 beads or the plate S-trap for proteomic digestion. Now, we also add some peptide quant before running the LC-MS injections to enhance reproducibility. and to inject a uniform amount. And we leverage the opentrons in this case to simplify the process and make manual pipetting much easier. And then we do the analysis on the with an LCMS system. In this case, it was the Orbitrap Exploris, and the data was searched in FracPipe, which was uploaded on Terra. And Terra is another scalable solution that we're using here at the Broad Institute, where we're able to upload many different software, and that allows us to run them natively on the cloud using computational resources. I want to emphasize here that we're using DIA, and that's why we need a scalable workflow to work with to search our data. Now, the first thing that we've evaluated when using the Covaris ultrasonicator, whether the conditions that we're using are sufficient to extract the most amount of protein from our tissue. And here what I'm showing on the left side is 3 different sonication parameters that we were using. 2 factors that we changed were the duty factor, which represents the total active time of sonication, but we've also evaluated whether we're using enough tissue lysis buffer. And as we see, by using 50% duty factor and 150 microliter of tissue lysis buffer, which shown in green is giving us the most amount of protein that's been extracted from the tissue. So we use that condition to process all of our samples in the subsequent experiments. And then the next thing that we've evaluated was peptide yield from these tissue. And the way we've designed this experiment was we started from 50 microgram of protein input, and it was either processed with the STRAP process in a plate format. And when I say STRAP with less than 2%, which is shown in yellow, that means that we immediately reduce and alkylate it in the Covaris plate and then precipitate it for the STRAP. Or with blue, we evaluated whether we need more SDS to evaluate the STRAP, as it requires 5% SDS per the manufacturers. And the last thing that we've evaluated is the common SP3 bead workflow, which was done by loading immediately also an SP3 from the Covaris plate. And when we look at the yields, we see the highest amount of peptides in the STRAP 2%. However, that was mostly shown in the colorectal cancer group, as shown in CRC among 4 different replicates. For breast cancer, we see peptides under 5 microgram, slightly on the lower side, and this has to do with tissue homogeneity and the nature of the tissue that we're working with. But overall, regardless of the choice, we found that STRAP works best for our case, as we can add another wash, which is the chloroform methanol wash during the process that allows us to further clean our samples. And the plate format does help us in keeping things as fast as possible. And then the other thing that we're showcasing here is the protein identifications from all these workflows. When we look at overall, we're seeing over 7,000 proteins identified, regardless of the method that was used. And that's consistent among 4 different replicates and in both breast and colorectal cancer blocks. So at this point, we've decided to use 50% duty factor, 150 microliter tissue lysis buffer, and then we couple that with a plate STRAP process without adding any additional SDS and just relying on the Covaris buffer, reducing and alkylating, then carrying on the digestion as normal as an optimal workflow to use. Now, some extra stats that I wanted to go over is the peptide depth. And again, we don't see much of a difference in terms of peptide depth. Another measure that we're looking at is total quantified tick from each experiment. We see reproducibility among all the experimental replicates and the tick that's quantified from each experiment, which gives us more confidence with the automation setups that we have in-house, but also with the workflow itself that's being reproducible when it comes to processing tissue and desalting. And last, we're looking at miscleavage rate for each workflow. Everything falls under 16%, which is also a good thing for a typical proteomic experiment. Now that we've optimized a workflow starting from full scroll analysis, what I want to go to next is how does this workflow behave in a tumor-rich environment? Many people who want to do, many researchers who are interested in biomarker discovery are interested in macrodissection or smaller regions where the tumors are enriched. So we'd like to see whether this trend would still hold true when working with smaller sections. So what we've done is we were using breast cancer and colorectal cancer blocks. And the idea was to macrodissect the tumor-rich areas. And in a macrodissection experiment, usually it can be lengthy, given that if you want to collect a full plate, one question would be how stable is a tissue in a lysis buffer? And for a histologist, it's preferable to transfer a tissue into a wet environment rather than into a dry environment so that the histologist wouldn't end up with any static related problems. So we've done an evaluation on the stability in the Kavaris plate, out of which 4 replicates from each block were split into 2 groups. One group was transferred into the plate with tissue lysis buffer in there, while the other group was transferred into a plate without any tissue lysis buffer. When the macrodissection was over, the tissues were transferred into -80°C, and they were frozen until time of analysis, which was carried on in the next few days using the STRAP process that we have in-house and some automation on the Opentrons. And we do stage tip desalting for sample cleanup, and we followed that up with a DIA acquisition on the Orbitrap Explorers 480 with a Vanquish NAIA. And what I'm showing here first is that when it comes to quantified protein yields from all of these tissue, we don't see much of a difference between whether having tissue lysis buffer or without in each well, which made us, which explains stability regardless of the tissue storage condition. And for our case, we prefer having the tissue lysis buffer in the wells when doing these experiments. Now, the other question that we had in mind is how does a depth change when working with full scroll with tumor-rich areas compared to full scroll analysis? And right here, we're seeing almost 8,000 IDs seen from all tissue. And that means that we're still achieving this deep proteome profiling while focusing these tumor-rich areas. And then when it comes to peptide depth, it's still within the 50K, which is expected based on our previous optimizations. One more measure of reproducibility is protein CVs. And what I'm plotting here is violent plots among the 4 different replicates from each different block. And as we see that the median CV for each protein abundant for all proteins across all blocks for the replicates are under 20%, which gives us more confidence in the reproducibility and show that there isn't much variation among experimental replicates. And one more question that we wanted to evaluate in a tumor-rich area is how efficient is the D-cross linking when we're using the Kavaris tissue lysis buffer and our digestion process. And one way to do this was to create a pooled sample out of which we've combined breast and colorectal cancer peptides. And these peptides were analyzed by a DDA, which is data-dependent acquisition, into different fractions. And we search that data in a PTM discovery mode, and that allows us to look for any potential modifications that we're not aware of. And you're no longer limited to the modifications that you're using in the database. And the results came back, out of which we see 80% of all peptidoforms that we've seen in this open search being artifact-free. And that is very good for a sample of such background where it's been sitting in formalin for many years, and we're able to recover a lot of these peptides. Another experiment that we were interested in is how does the quant behave with this workflow when we're using different instrumentation for DIA, but also how does it compare to the TMT, which is multiplexing, which is a very common strategy used in proteomics. And we also wanted to use some ground truth, such as a positive control here. And one idea was to use a genetically engineered mouse model to evaluate the FFPE sample processing. So in this experiment, we're starting with 4 different blocks. 2 blocks contain organs that were embedded from wild-type mice without any without any modifications. And then we had 2 other blocks that originated from genetically engineered mice that had an overexpression of the NCOA4 gene. And we processed these samples. We've scrolled these blocks into 4 different replicates. And then the idea is to pass it through our FFPE workflow. And then once we reach the peptide level, that's where we split the sample for different type of mass spectrometry analysis. One workflow would be using the TMT acquisition process, where individual samples get individually labeled with different tags. It gets pulled again into a master mix. That master mix gets fractionated into 24 different samples, and then analyzed by LC-MS2 on the Orbitrap Explorers 480. The data gets searched in FracPipe and MS-Fragger. And even though this process is very lengthy, which takes about 7 days, it still acquires at a slower pace compared to DIA at 8 samples per day. And then the other thing that we were evaluating was how does DIA behave compared to TMT. So we've taken each individual sample and we've desalted it into HPLC vials. And these samples were cleaned up immediately and injected into 3 different instrumentation. The Orbitrap Explorers, where we're using the wide window DIA in a variable scheme, the timsTOF HT, which uses the ion mobility to further enhance our reproducibility, but also enhance the proteome depth of what we're working with. And we're also using the newly released Orbitrap Astral, which is all known to be very fast and also with high resolution. We were able to use narrow DIA on that as well. And the data was all searched in Diane, which was also uploaded on Terra. And it's worthy to mention that the process of DIA would require a sample prep time of only 2 days compared to TMT, which is 7 days, while the LC-MS acquisition time is at least twice as fast when you're using an older instrument like the Explorers 480 at almost 12 samples per day, while the timsTOF HT can catch up to almost 30 samples a day, and then the Orbitrap Astral is able to sequence at 60 samples per day. And all of this compared to TMT at 8 samples per day. Now, the first thing we want to look at is how does the protein depth change between TMT and DIA? And surprisingly, we see Orbitrap Astral almost catching up to the depth that's seen by TMT, even with the extensive fractionation of 24 different samples. Now, with the timsTOF HT, we're seeing almost 8,500 proteins as well, and the Explorers 480 is still at 8,000 proteins, which is still pretty impressive for the method that's still faster than the TMT. One more thing to look at when we're working with proteomic applications is peptide quant and peptide identifications. As expected, TMT would give us the deepest coverage when it comes to peptides, given the deep fractionation. However, we see Orbitrap Astral also catching up at 90,000 peptides. And that validates the use of DIA for proteome depth, as we can cut down sample prep from almost a whole week to 2 days, but also speed up the acquisition time to 60 samples a day. And we see the timsTOF HT and the Explorers 480 still giving us reasonable peptide depth of 70 and 55 peptides. The other positive control that we were interested in is the NCOA4 gene. And we do see it overexpressed in all of these samples when compared to the wild type. And what I'm plotting on the right side is the full change assessment of the 4 different groups. We do see, though, the TMT, which is shown in purple, underestimating the protein full change of NCOA4 that was measured by old DIA method. That's shown in the green shades. And I will get to that in the next slide, as we know that TMT is relying on the concept of multiplexing. So it can go through a process where interference can happen between different channels, and that would suppress the observed fold changes. So what I'm plotting on the left side here is a correlation matrix where I'm showing how do all measured fold changes from all these blocks compare between Astral with respect to TMT, the timsTOF HT, and the Explorers 480. And as we see on the y-axis where the Astral is plotted, we see that both DIA methods, which are shown in the green shades, they align very nicely in terms of fold changes. and exhibit Pearson correlation over 90, which is pretty strong. Now, when it comes to TMT, we do see that the Pearson correlation drops slightly to 0.8, and that is expected given the signal compression problem that I just described of TMT. And one more metric that we're looking at here among the experimental replicates is the protein CVs for each method. And as we see TMT shows the least amount of variation, and that is expected because you're multiplexing and you're doing relative quant. But we see DIA also being reasonably good, and all of our CVs are contained under 10 or 20%. And one last thing that I want to show here is how does NCOA4 is NCOA4 a significant hit in our experiment? And the volcano plot here shows us that NCOA4 is significantly upregulated, as shown by the volcano plot, but also we see some other biomarkers that I have plotted in here. And that basically serves as like a ground truth for this workflow, given that we know that this gene is overexpressed genetically, and we're able to recover that piece of information out of FFPE blocks within a reproducible manner while achieving good depth and very fast acquisition speeds. So I want to go over what we've done so far. Really, the Covaris ultrasonicator have really simplified the problem of processing FFPE samples, especially eliminating the need to use something as toxic as xylene. And that sped up the process significantly, especially when we're working with 96 well formats. And then this workflow enables tissue to be processed real fast. And we expect results to be out in almost 5 days, given the speed advantage that we have now with both instrumentation and FFPE tissue processing by ultrasonication. And the best thing is that this workflow is compatible with different instrumentation. So you don't really need the most recent instrument to get good depth, as the sample prep has been really optimized to give you reasonable and impressive depth from regardless of the instrument that you're using, as long as you're using label-free analysis. And then last, I really want to acknowledge many people that got involved in this project. From the Broad, really, Shankar, John, Simone, Mike, and Mani, and Stephen Carr. From the Covaris team, we've received a lot of help in terms of diagnosing issues and getting the authentication conditions correctly, setting up the instrumentation and all the assistance that we needed to build this workflow. And I want to give special thanks to Lillian from Thermo for helping us demo the Orbitrap Astral as well with these samples, and for the Van Andel Institute for helping us with a lot of the macro dissection that we've done of these FFPE scrolls. And of course, Govindan from Washington University in St. Louis. And we'll take it to questions from here.
Mo, thank you so much for that great presentation. And Deb, you too. It is now time for our live Q&A portion of our webinar. So to our audience, if you haven't submitted any questions yet, please do so now. Just type them into the Ask a Question box and click Send, or click Submit, and we will answer as many of your questions as we have time for. Mo, Deb, we have quite a few questions already coming in, so I'm just going to dive in. And Mo, let's start with you. This question is 2 parts. How was the optimization process in working with Covaris to determine the ideal duty factor and volume settings for your FFPE workflow? And can you please describe what the optimization process was like?
Covaris has provided us with standard protocols where we've evaluated our tissue and we've tried these first. However, we were in direct communication with the Covaris team, and the way to optimize the amount of protein that was extracted from each sample was in tandem with our communication with Covaris. What we've done was looking at the duty factor and the volume settings, as we did not see complete homogenization of FFPE scrolls, and that is expected given that tissue can be different and blocks can be of different dimensions. So what we came up as a result here was 50% duty factor and 150 microliter of tissue lysis buffer was optimal to achieve the greatest protein amount of protein that was extracted. We've also coupled that with some ID identification evaluations for both duty factors, and we didn't see much of a difference. So it's a balance of both sample preparation and the optimization as well on the sonication side.
Great. Thank you, Mo. And I'm going to get this next question over to you. You mentioned scalability as one of the benefits when using Covaris. Have you thought about taking that further and automating your FFPE workflow?
Yes. So for now, we're using we do some semi-automation by relying on the OpenTron's automated liquid handling. And for many people that are involved in label-free analysis, and especially with tissue of such background, it is challenging to do a lot of manual pipetting in a 96-well format, especially if you have to deal with different volumes. So what we do is the OpenTron helps us significantly by using custom CSV input files where we would assign what volume is going into each well. Now, the other thing that we also do is we use the plate-based S-trap so that we can speed up the digestion and we'd be able to get away with multi-channels. However, I can see that this workflow can be fully automated if it needs to go down to that path with both Covaris and some robotics on the side.
And Mo, I'm going to throw another one at you. This one has a few parts as well. How do you think Covaris workflow compares to the traditional FFPE extraction methods? Is it easy to follow? Does it require a lot of training? And were results consistent end to end from end user to end user?
Absolutely. The traditional workflow relies on xylene, so that's already a toxic chemical to work with. But also, it's much more manual work compared to what we're doing now. Being able to process and transfer tissues, transferring tissue right into the plate is really helpful here. And for anyone who's macro dissecting, it's much easier to just take the regions of interest and then transfer that into a wet environment, which is the Covaris lysis buffer. And then right after the process is complete, you would just seal that plate and store it at ADC. You've eliminated days of work by just using the Covaris setup rather than doing the dewaxing and homogenization of each sample individually.
Thank you, Mo. Deb, I'm going to jump over to you. Can your workflow also be used for intact protein purification?
Yeah, that's a good question. And the answer is yes. Ideally speaking, if the protein is not necessarily cross-linked into a certain matrix, you can potentially use the technology for unhooking or extracting the proteins, and depending on the buffer of choice, you can actually put it in a non-denaturing buffer so that you can ensure its native, if not intact structure. If it is in a matrix like FFPE, where it's cross-linked, when you decross-link it, it's more than likely that you are not going to retain its native structure. But if you're talking about intact proteins, yes, only the buffer section will have to be different necessarily, and you do not continue forward with the whole workflow of purification and digestion.
Thanks, Deb. Now let's talk about kits. Can the kit components or methods be optimized to accommodate higher starting amount of type of protein?
So these kits that I showed in my presentation will not necessarily accommodate something that is significantly different or higher, but we do have workflows. I also showed one particular slide that talked about same type of matrix, but different numbers of cells or different amounts of fresh frozen tissues. So we can address different input amounts with different workflows. The kits per se ask for a range, like our cells talk about anywhere between 1,000 to 500,000 cells. If somebody's talking about doing 2 million cells, it's probably going to go to a different workflow, which we can also support.
Great. Now, Deb, we have a few questions similar to this, and we have some attendees considering several kits and workflows that exist for protein purification. Where and how does your AFA-enabled workflow work better?
Yeah, I mean, that's a very overview kind of an answer that I can think of. That, to begin with, most of the extraction protocols that are there, and most data also testify to that, that most of the extraction protocols focus on getting proteins out without paying attention to, it's like a one size that fits all kind of an approach. So it doesn't necessarily say that, oh, I don't need to hit it with this much of power if I'm trying to lyse cells versus if I have an FFPE sample or versus I have a skin tissue. So that's where the ability of controlling the power, which you get when you're operating with an AFA technology, and then the whole workflow gives you better, specific, optimal functioning capabilities to extract as much as you can of the proteins, followed by the one plate capability where you can actually do the whole workflow of protein extraction, purification, and digestion in the same plate, and the expedited features. Those are all unique capabilities and unique benefits which you're enjoying from a non-invasive energy.
Thank you, Deb. Now, is the kit suitable to digest peptides and proteins without changing the chirality of the amino acids?
Is the kit suitable to digest peptides without changing the chirality of? We have not necessarily tried or tested it, but because essentially it's looking at protein digestion. So it cleaves using the very standard enzymes. So if you are looking at the same lysine arginine residues that trypsin would go after, I would say it wouldn't necessarily cause any significant changes to the chirality. We haven't really gone into the depths of looking into small peptides and digesting them using this because that is not necessarily what most people do.
Yeah, we're past our time, but I want to squeeze in a few more questions. So, how does AFA enabled digestion ensure better reliability of digestion?
So the hypothesis is that the low power pulse during the time of digestion incubation actually opens up the hydrophobic core of the proteins, making it a little easier for the enzymes to have access to the hydrophobic cores. So that way it can actually do its job while it is still being active. And it also, in a sense, it enhances the whole process. So that's the hypothesis, and that's exactly what the micromixing is supposed to be doing. So, in other words, these on-bead or off-bead digestions ensure not just the speed of digestion, but also ensure that the enzymes do not necessarily cleave at the wrong sides. And there is enough tumbling of the molecule during homogenization that exposes the right places for digestion.
Thank you, Deb. And let's wrap with this final question. Do you also have established workflows for fresh frozen tissues?
Yes, we do have workflows for fresh frozen tissue. One of the slides in my presentation actually showed that. We do have fresh frozen tissue where we actually have specific consumables also, which comes in with a bead, with one bead that is very capable of actually addressing up to 10 milligrams of tissue and homogenize it using one of our flagship instrument and going through the whole process of purification followed by digestion. So we do not necessarily have a kit for fresh frozen tissues, but we do have workflows that work for multiple different types of tissues.
Thank you, Deb, Mo, for this great Q&A. I do want to thank you both for your time today and for your important contributions. Do either of you have any final comments for our audience?
I hope everybody enjoyed the talks, and I particularly enjoyed listening to Mo. So it was very insightful, lots of amazing data. I hope everybody also enjoyed the talks and also have a few thoughts to take back with them as to when they face their next complex matrix and they are trying to get proteins and peptides out of it, that they would think about, innovative, creative ways by which they can maximize efficiency and throughput.
Yeah, and I'll add to that. I mean, it's really exciting to see that we're able to process FFPE tissue in such a high throughput fashion. We're really excited to see what biology we're going to uncover out of this workflow. And please reach out if there's any questions.
Gentlemen, thank you both so much again. We also want to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webcast. Now, before we go, I do want to thank our audience also for joining us today and their interesting questions. We had quite a few questions that we weren't able to answer due to time constraints. So those questions, additionally, along with the on-demand questions, they will be answered via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. To all our friends out there, thank you for joining us. Have a great day, everyone. Bye-bye.
Thank you.